Okay, this the third period. I think I will make a bigger effort to finish this lecture around 12 o'clock. So you're able to bear with me for another 70 minutes. The content is rich. There's tremendous amount of information which I need to convey to you. And to able to study this microbiology well, you need to read the textbook. And so the PP is essential reading, but because this is class, so the more material is in the textbook. So according to the textbook given by your teachers, you can find the textbook in your library or wherever you can find. And if you read through the relevant chapters, then you will get more memory and get more understanding. So this is quite important because this is a huge science, and without putting more effort into it, then we cannot get much understanding, and we'll cannot get much memorization. That is, anything, any discipline needs learning, understanding, and memorization. Okay, now let's first talk about the cell wall structure. That is the structure outside of the plasma membrane. So this is an important area, difficult to understand. So this is schematic, again, a schematic diagram of the two kinds of bacteria. That is gram-positive and gram-negative ones. Okay, on the left side is the gram-positive ones. It looks simpler because it has one plasma membrane, also called the cytoplasmic membrane. And outside of it, we can see, you can find peptidoglycan. Peptidoglycan is generally thought as the cell wall. And also the outside is capsule. Capsule is sometimes present or sometimes not present, but it's very important in virulence factor or pathogenetic factor. That is, this part is connected with the pathogenicity of this particular bacteria. And on the right side is the gram-negative bacteria. Gram-negative ones, we can see, much more structures. That is the outside of cytoplasmic membrane. There may be the peptidoglycan. Peptidoglycans here, the very thin peptidoglycan. And another layer, it's called outer membrane. On the outside outer membrane, there's another, there's a capsule or other structures. And so there's a space between the inner membrane and outer membrane. It's called the periplasm. And so it's it has many more structures. Many more structures. So I'll give a more detailed description of these gram-positive, gram-negative ones. First, let's talk about the Gram staining. This person, Dutch scientist called Christian Gram, in about 1880 something, 1880s, he invented the staining method. And this staining method can distinguish two great categories of bacteria. One is called gram-positive, one is called the gram-negative ones. And the gram-positive ones, you can see that the final stain is purple, bluish colored. And for gram-negative ones, it's pink, reddish colored. Okay, so it's the staining is divided into 4 simple steps. The first is crystal violet. Crystal violet is a violet dye. The color is violet, deep blue colored dye. So apply this dye to the cells. The cell has to be fixed, that is killed on a slide. And then wash the dye off. After drying in the air, then apply the gram iodine. Gram iodine is the iodine solution. It has iodine, I2 and the Ki, I2 and the Ki solutions. It's called a mordant. Sometimes it's to fix dyes called mordant. And then after this 1 minute, this is 1 minute, and after cleaning this another 1 minute, and then clean with tap water. And then the third step is decolorization. Decolorization is alcohol or acetone. That is. And this is 95% alcohol and pure acetone. So after this step, there's a tremendous difference. That is, this is totally destained. That is, no color is retained. But this still retains the color. It's purple, bluish purple. And then finally, the fourth step is called the safranin red. This is a pink, reddish color dye. And use it to stain another minute, and then wash under the tap water. That is, after every step is 1 minute. And after the staining, the dye or the solution will be removed by tap water, rinse in the tap water. Okay, then this retains the pink color, red color, and this retains the blue color. So this is a very convenient way to do, because it takes only about 10 minutes to do the staining. And also we can distinguish 2 kinds of bacteria. One is the blue colored, one is pink colored. Blue colored is called gram-positive. Pink colored is called gram-negative. Okay, so these are the great division of bacteria into gram-positive ones and gram-negative ones. Okay, so this we need to see the picture here. You see the pink ones here are gram-negative ones. The dark ones, blue ones, are the gram-positive. So this is a slide containing a mixture of gram-negative ones and gram-positive cells. Okay, so what's the difference? The difference is in the structure of the cell wall. So the gram-positive ones are simpler. So the outside the thick peptidoglycan wall. That is thick. Thick is a thick of up to 80-70 layers of the cell wall. And inside of it, there are many second kind of molecule is called teichoic acid. Teichoic means wall in Greek. So teichoic acid means wall acid. Lipoteichoic acid means this teichoic acid is linked to the lipid. It's called lipid lipoteichoic acid. So gram-positive bacterial cell wall consists generally of these 3 kind of molecules. First is peptidoglycan. Peptido means peptide means a small chain of amino acids. Glycan means a sugar chain. Teichoic acid means wall acid. Lipoteichoic acid means lipid wall acid. Okay, let me explain. Generally, the gram-negative ones. Gram-negative ones is more complex. That is, you see, the inner plasma membrane, the outer plasma membrane, outer membrane. Then in between, we have periplasmic space or periplasm. Peri means around it, around the surface. It's called periplasm. Periplasm generally is about 20 to 40 nanometer wide. So the total space is about 40% of the cell volume. And it has some molecules in it. various kinds of molecules. So this is the nutrient binding proteins, also peripheral protein. So this is the pore proteins stuck inside the outer membranes. And also this is very thin peptidoglycan. So the difference here is important because the gram-negative bacteria is more complex in its cell wall structure than the gram-positive bacteria. Okay, so these structures will be explained in more detail later. Let me give you a electromyograph of the gram-positive cell surface and gram-negative cell surface. So this may have some shrinkage. So you can see that it is a rugged surface for the gram-negative ones, and less rugged surface for gram-positive cells. Okay, let me explain the structure of the gram-positive and the gram-negative peptidoglycan. Peptide is here. Peptide. This is only a straight chain, but this is a cross-linked. This is cross-linked, and this is the straight chain. This is glycan. Glycan means a polymer of sugars called the glycan. This is a glucan, galactan. Those are large polysaccharide molecules can be called glycan. So you have learned in biochemistry. Have you learned biochemistry? So biochemistry is another very important science that you have to learn. Okay, so these are the very simple diagram of the glycan as a straight line, straight chord, and then the peptide is a cross-link. Okay, now let me explain in more detail the structure. The glycan is two sugar unit. It's called N-acetylglucosamine. It's called N-acetylmuramic acid. Okay, here. Let me show you more detailed structure about the sugar. Sugar here. These are the more detailed sugar structure. N-acetyl means this is amino sugar. Amino sugar is here. Here's the amino sugar. That is amino group here. But this amino sugar has one acetyl group. So this is called N-acetyl glucosamine. This is amino sugar here. Acetylglucosamine. And then N-acetylmuramic acid is here. You can see this is almost the same structure here. N-acetyl, like glucosamine, right? But here, it has one lactic acid. So with the lactic acid is called N-acetylmuramic acid. Mura is a wall. It's also Greek word for wall. Teichoid is, I think, Latin. Mura is Greek. Western science use a Greek name or Latin name to the scientific name. So this muramic acid is also a wall acid. Because of this structure, this lactic acid, it can link to a peptide. So this peptide is called tetrapeptide. It's 4 amino acid. Originally it was 5 amino acid, but after linkage it became shortened. It became 4 amino acid. So this is we can see this is alanine, and this is alanine. Okay, it's a glutamic acid. This is methyl. Methyl means there is no D at all. It's a diaminopyramidinic acid, and And this is called pimelic acid. This is similar to lysine. Lysine has four C, 4 carbon and 1 amino group, right? This has 4 carbon and 1 carboxyl group, 1 amino group. So this is a little bit different. You can see that this is aeroform amino acid, but this is different. It's also different. So the bacteria uses deformed the amino acid to construct this tetra or penta peptide. So this is a very most detailed structure, most detailed structure of the sugar by sugar unit. That is acetylglucosamine is called the G, abbreviated as the G, M here, and then from the M, from the lactic acid, then short peptide chains formed. Then out from this place, this amino group will form a peptide chain, peptide group, peptide bond with another chain from another glycan molecule. So this becomes a cross-link. So this cross-link is very important for the strength of the peptide glycan, because the longitudinal line is the glycan, and then the horizontal linkage is the peptide chain. It's peptide chain, and so this became like a fabric like a cross with the longitudinal lines and the horizontal lines, and so this became a strong bag. Okay, let's go back to the previous structures. Negative. Okay. Okay, after displaying the detailed structure of the glycan and the peptide, then I explain the difference between gram-positive, gram-negative peptide or glycan. There are minor differences between the gram-positive ones and the gram-negative ones. Each bacteria has its particular variations, and so we cannot remember all of this. But because the E. coli as a standard gram-negative ones, and the Staphylococcus aureus is a standard gram-positive ones, their structure is solved in more detail. So we only need to know their differences. So Escherichia coli is the colon bacteria, one of the major colon bacteria, and it's gram-negative ones. It's in our colon. Staphylococcus aureus is a golden. Aureus means gold. Golden Staphylococcus, golden grape-like bacteria, and it's generally on the skin, in our throat. So it has many pathogenic properties. You can see that the backbone is generally the same, right? But however, for the gram-negative E. coli, the linkage is from here. Diaminopimelic acid, it links to the alanine, and this alanine was originally has another alanine, that is Ella-glu-dap Ella. Once it forms a linkage, then this alanine was cleaved. It's cleaved, and And this cleavage is important because it cannot form another linkage. Okay, cleaved. So this is a direct linkage between the two short peptide. It's indirect because it has an interbridge. It's called the pentaglycine. Glycine is 5 glycine bridge linking this lysine to this short chain. So there is an interbridge here, but there is no interbridge here. Okay, during this process, there's a enzyme outside of it to promote the linkage of one peptide to another one. It's called a peptidase. And this peptidase has one inhibitor. This inhibitor is penicillin. So penicillin, once binding to this transpeptidase will inhibit the transpeptidase. So in the presence of penicillin, the bacterial cell wall cannot be cross-linked, so it's weak. So it will burst open. So cannot live. And so this is the mechanism of antibiotic penicillin. So all sorts of penicillin-like family antibiotics works by inhibiting the transpeptidase. Okay, so this is the major features of the peptidoglycan. And then let me show you other structures in the bacterial gram-positive ones. Okay, by the way, let me say penicillin inhibit the bridge formation. Lysozyme, which can cut. Sorry. Cut between the chain. This is beta-1,4. Beta, this is 1, right? And this is 4. From 1 to 4, beta-1,4 glycolytic chain, glycosidic chain. This glycosidic bond will be cleaved by lysozyme. Lyso means dissolve, zyme means enzyme, dissolve enzyme. Was discovered by Fleming. Fleming also discovered penicillin. Before he discovered penicillin, he liked to play with things. So he used his saliva, used his tears, used his nose drippings, and to try to see if it can kill bacteria. And he did find that human secretions, even egg white, has this enzyme. This enzyme can kill bacteria. So he purified this and named it lysozyme. That is, it can kill bacteria. So lysozyme is actually one immune defense proteins in animals, and it can protect cell outside, especially from the mouth, the nose, the tear. From its presence, it can kill any bacteria which are trying to get into human body through these places. So this is a very important natural defense proteins called the lysozyme. Protoplast is a wallless cell. That is, if we treat the cells with penicillin, but in an isotonic solution. So bacteria will not burst open because it is outside, the inside is of the same osmotic pressure. So they will not break. So when I break, then so it became a wallless cell. So this is called a protoplast. Okay, so I have slightly explained the structure of gram-positive cells. And now I go into the gram-negative ones. Gram-negative ones is more complex. You can see that this is really complex. Okay, the outer membrane has many, many proteins. Some proteins are called porine. Proteins are three-barreled, two tubes form a protein, which will allow some small molecules to pass from outside to the periplasm. Okay, so it has a selective passage for the small molecules. And also it has some proteins for various functions. Okay, and also has this structure and this lipopolysaccharide structure. Now let me explain this structure. Structure very, very important. Lipopolysaccharide. Okay, so this is lipid and also polysaccharide, also called endotoxin. Okay, also endotoxin. Okay, so it is called endotoxin because once the bacteria is in the cell, inside the human being, inside an animal, then the release of this endotoxin, well, is sometimes lethal. They cause septic shock, or called bacterial shock. Bacterial shock means that the bacteria gets into the bloodstream, and then the lipopolysaccharide was released. And this lipopolysaccharide is a very strong stimulator of the immune response. It induces the innate immune response. That is, the immune cells in the blood will be angry and they will release a lot of cytokines to cope with the LPS. At the same time, cause the patient to fall into an unconscious state. That is, their organs are working less well, and their blood pressure is going down, and the people is unconscious, and with no emergency help, then this person will die. So this endotoxin in the animals' circulation will cause septic shock, or called bacterial shock. This bacterial shock is generally the main cause of bacterial disease death. But in the cell, gram-negative cells, it has many functions. First, it increases negative charge. Also helps cell to adhere to the surface. Okay, and also induce the strong response from animals and also bacteriophage, sensitive to bacteriophage. So it has its own function in the bacteria. However, because the animals are fighting this bacteria for over millions of years, and so animal cells are very sensitive to LPS. So if bacteria gets into the bloodstream and release this LPS, then the patient will get into the septic shock state. Okay, so this is the importance of the lipopolysaccharide. So lipopolysaccharide is embedded in the outer leaflet of the outer membrane. Let me show the structure of it. So this is also very important. Then the LPS is this structure. That is, this is the lipid structure, right? These are sugars. This is the phosphate sugar, diphosphate sugar. And then this is the polysaccharide. And this polysaccharide also has outer polysaccharide. It's called O antigen. O is in German, it's the body antigen. The cell body antigen is called the O antigen. And then this core antigen. Core means it's a repeated unit. Well, not repeated unit, it's a unit, just invariant unit. But this is invariant in the length. That is, this circle is a group of sugars. It can repeat many times. So the whole antigen can be many sugars long. But this one, core, is the 7 to 8 sugars long. And this part structure, okay, it's amine. It's amine lipid. Amine lipid means that this is glucosamine, and then from the amino group, this is linked with the fatty acid. So this part is called the lipid A. The toxin, the endotoxin, the major ingredient, major part of the toxin is this lipid A, and the outer part is not toxic, but it's generally is linked with the lipid A. So the whole structure, LPS, is very toxic. Violent symptoms. So this is a very important part in the gram-negative cell wall. Very important. Okay, so I have just explained briefly the gram-negative cell wall. Then let's go back to the gram-positive cell wall. Gram-positive ones, that is, it has many layers of peptidoglycan and also has two-wall acid. This is called teichoic acid. This is lipoteichoic acid. Teichoic acid is also an acidic acid structure like this structure. That is, it's either glycerol with phosphate or ribitol with phosphate. Ribitol is a sugar alcohol, 5-carbon sugar alcohol. 6-carbon sugar is glucose, right? In galactose, that is fructose. And 5-carbon is ribitol, ribose, or arabinose. So those are the 5-carbon sugar. So this is a sugar alcohol called ribitol. So it has a 5 OH, right? Here, OH. 2 of these are linked to phosphate, and the 3 of these are linked with some amino acid. So this unit is repeated many times. So this is a phosphate, sugar, phosphate, sugar, phosphate, sugar, maybe 10 times or 20 times. So it became very long. So the outside is many phosphate. So this increases the acidic property of the cell surface. That is, the bacterial outside is generally acidic, that is negatively charged. Okay, so this is either glycerol phosphate or ribitol phosphate repeated many times, became the teichoic acid. And this teichoic acid is linked to the lipid. It's called the lipoteichoic acid. Okay, so I have tried to tell you the intricate structure of the cell wall. So hope that you can learn this part well. So this is another diagram of the LPS. This is amine ester, and this is a core polysaccharide. This is O-specific polysaccharide. Okay, so let me summarize the gram-positive and gram-negative outer membrane. That is, it has the gram-negative ones has lipopolysaccharide, lipoprotein, phospholipid, and also these structures. And you can see from each micrograph, the gram-positive ones are simple. That is the one thick wall and one plasma membrane. Okay, see, one plasma membrane, thin plasma membrane, and a thick peptidoglycan. But for the gram-negative ones, we can see a few layers. One outer wall, one peptidoglycan in the middle, and one cytoplasmic membrane in the inside. So these are the differences between gram-positive ones, gram-negative ones. Let's go back to explain why gram-stain is able to distinguish the gram-positive ones and gram-negative ones. Okay, the crystal violet will link with iodine, to form larger crystals, larger complexes. And this one, that is when alcohol is used to dehydrate the peptidoglycan. Peptidoglycan gram-positive cells are thick. So although alcohol shrinks the pores because of these 80 layers of the peptidoglycan, so the holes is very small, very small. So crystal violet iodine complex cannot leave because this molecule is larger than the pores on the wall, on the peptidoglycan. However, for gram-negative, because its peptidoglycan is only a few layers, and so once alcohol treated this peptidoglycan, with the dissolution of the plasma membrane, and peptidoglycan is still a large pores, so crystal violet and the iodine complex leaks out. So this makes gram-negative bacteria cannot retain the crystal violet and the iodine complex. That explains the different staining properties of gram-positive and gram-negative bacteria. Okay, let's struggle for the last 30 minutes. Okay, now the archaea, which I will introduce in the third lecture, is a new domain of bacteria, which is different from the bacteria in many ways. And its cell wall is quite different. So it has called a pseudopeptidoglycan. Pseudo means force peptidoglycan, because it is not muramic acid, it's N-acetyl-talosamine-muramic acid. That's a different kind of sugar instead of N-acetyl-muramic acid. Also, its linkage is beta-1,3 instead of beta-1,4. So lysozyme cannot dissolve, kill cell wall. N-acetyl-talo. There are some bacteria who do not have the cell wall. Protect environment, and some archaea is also okay. As for the archaea, the plasma membrane, the phospholipid is here. Phospholipid is here. This is phosphate, and this linkage is ester linkage. Ester. COO is ester linkage. This is phosphate, glycerol, but it's ether linkage, so it's very different. So this difference, it has a long penta unit, 5-carbon unit. It's called isoprene unit. So in total, it's 20 carbon candy carbon unit. So this is called the it's called the glycerol diglycerol tetraether. Two not a two-layered membrane. It's single one leaf. Plasma membrane is one leaf only, instead of two leaves. So this can express resistant. That is a thermophile. It's much more thermophile. So these are the lipid single layer. These are archaea cells. Okay, so I have tried in much more detail. And now let's go into other structures. The first is the inclusion inside or on the periphery of the bacteria. One is called velutin. Velutin is called metachromatic granule. Metachromatic means than the general cell color, so called. And also velutin means inside